Hey everyone, this lesson is on chloroquine and hydroxychloroquine's mechanism of action, more specifically on its antiviral properties against SARS coronavirus 2 or SARS-CoV-2, the novel coronavirus that causes COVID-19. So if you want more information on chloroquine and hydroxychloroquine in general, please check out my other lesson on that topic. So chloroquine and hydroxychloroquine have multiple effects on cellular and organelle functioning. For one, these medications have immunomodulatory effects. They can actually inhibit certain immune cell functions, which is helpful when we're trying to treat autoimmune conditions. So they can actually be used to treat certain rheumatological conditions like lupus and rheumatoid arthritis. I'm not going to talk about these immunomodulatory effects in this lesson, but if you want more information, please check out my other lesson on chloroquine and hydroxychloroquine. The second major property that these medications has is that they can alkalinize vacuolar or organelle pH, like the lysosomal pH. So the reason is that chloroquine is alkaline, and it can actually enter into organelles like the lysosome, and they can enter into endosomes as well to actually increase the pH of those organelles, decreasing those organelle function. So it can be used to treat malaria. So malaria is caused by the protozoa of Plasmodium species. So it can inhibit protozoal food vacuole functioning by alkalinizing that food vacuole. And as I mentioned before, it can actually alkalinize the endosomes, lysosomes, and it can actually inhibit endocytosis and lysosomal fusion and function. The third property of chloroquine is that it is known to be a zinc ionophore. So being a zinc ionophore, it can actually allow the influx of zinc into cells and into organelles like lysosomes. And there's some question as to allowing the zinc into cells, does it also have an anti-cancer effect as well? I'm not going to talk about that here, but that's something interesting to note as well. And the fourth point I want to make here is that chloroquine and hydroxychloroquine are known to bind to sialic acids. And I'll talk about what sialic acids are a little later on. So point 2, 3, and 4 are the points I'm going to talk about in this lesson with regards to antiviral action against SARS-CoV-2 or the coronavirus that causes COVID-19. So a quick disclaimer before I get into the antiviral properties of these medications. This evidence I'm going to show you has been shown in vitro studies, essentially in a cell culture petri dish. So keep that in mind before we get into these antiviral mechanisms of action. So before I get into the mechanism of action, I want to lay down the setting as to where all of this is taking place. So here is the plasma membrane of the host cell, and here is the host cell itself. Here is a lysosome, an acidified organelle that is responsible for recycling of nutrients and other waste products. And here is the nucleus that contains the genetic code. The first point I want to talk about here is that chloroquine inhibits endocytosis. We talked about how chloroquine is an alkalinizing agent. Well, we're going to talk about how it blocks endocytosis. And there's a paper that has shown that it can block Zika virus infections. So this is where we're going to start. So this paper talks about how it blocks endocytosis. So the SARS-CoV-2 virus is here, and this virus contains spike or S proteins on its surface. These S proteins allow it to bind to ACE2 receptors on the host cell. When it does actually bind to the ACE2 receptor, it can actually be endocytized into the cell, into an endosome. So it gets brought into the cell in this little endosome. That endosome will eventually transverse through the cytosol and fused to a lysosome, where the virus can enter into the lysosome and exit the lysosome, allowing it to infect the cell. So with regards to chloroquine can block this process through entering into the cell, and it can actually permeate into endosomes and the lysosome, allowing it to alkalinize the endosome and lysosome, which means that it can increase the pH of the endosome and lysosome, preventing it from being acidified. So those lysosomes and endosomes are not acidic the way they are supposed to be. So they become less functional or dysfunctional. It may also inhibit a proton pump on the lysosome. I've talked about this in previous lessons that there may be some inhibition of a proton pump on the lysosome known as VATPase or vacuolar ATPase, although this is not entirely known at present. Nevertheless, due to the alkalinization of endosomes and lysosomes, endocytosis, the process of endocytosis, bringing the virus into the cell, is actually inhibited. It's stopped. And the fusion of endosomes to lysosomes is also stopped. So this whole process whereby the virus can enter the cell and get in can be stopped or slowed down. So the way this happens again is that chloroquine increases endosomal and lysosomal pH, and in doing so decreases endosomal and lysosomal functioning. So it decreases or inhibits endocytosis. So the second point I want to make regarding chloroquine's antiviral properties is that chloroquine is a zinc ionophore. There's actually a paper published in PLOS ONE in 2014 indicating and showing evidence that chloroquine can act as a zinc ionophore. That means that chloroquine can allow the entry of zinc into a cell. So how does chloroquine actually do this? So without help, zinc cannot enter into a cell. It essentially bounces off of the cell membrane. But with chloroquine, it can actually attach to chloroquine, and chloroquine can actually guide it through the plasma membrane, allowing zinc to enter into the cell. So why is all of that important? So the reason that all of that is important is that there's a study showing that zinc inhibits coronavirus and arterivirus RNA polymerase activity in vitro, and zinc ionophores block the replication of these viruses in cell culture. And this paper was published in 2010. So normally, when a virus enters, specifically an RNA virus, there's an RNA-dependent RNA polymerase, or RDRP. That RNA-dependent RNA polymerase will essentially make viral RNA. There's a lot of steps involved, but I won't get into all of those steps here. Nonetheless, this RDRP will produce viral RNA. It replicates the virus. However, when there is chloroquine and zinc around, these can cross over the plasma membrane together. Again, the chloroquine acts as a zinc ionophore, allowing zinc and chloroquine to enter into the cell. We talked about this before. Chloroquine has its effects on the endosomes and lysosomes, but this mechanism here more specifically has to do with the zinc. So as the chloroquine brings in zinc, the zinc concentrations in the cytosol increase, and the zinc can then act to inhibit this RNA-dependent RNA polymerase action. So it prevents that polymerase from making viral RNA. So it essentially stops the virus from replicating its genetic code. So it stops making viral RNA. So this study that I just showed actually shows that as zinc concentrations increase, viral RNA decreases. If you want to check out more on that, please check out that paper and look at some of their data. They show some interesting data. So again, as zinc concentrations increase, viral RNA decreases. That is the second mechanism. And the third point I want to make about chloroquine and hydroxychloroquine's antiviral properties is based on this newest article that is being published in April of 2020. So this article is entitled "Structural and Molecular Modeling Studies Reveal a New Mechanism of Action of Chloroquine and Hydroxychloroquine Against SARS-CoV-2 Infection." So what does this paper show? So it all has to do with the cell membrane around the ACE2 receptor. There's actually something called sialic acid that is attached to the cell membrane that is usually pretty close to the ACE2 receptor, although it can be on much of the cell membrane itself. Sialic acid is essentially just a little piece of sugar, and the spike protein, the S protein on the SARS-CoV-2 virus, can actually bind to the sialic acid as well. So these sialic acid residues actually can act like little receptors for the S proteins on the coronavirus as well. So what this paper shows is that chloroquine and hydroxychloroquine can actually bind to these sialic acid residues, preventing the S protein on the SARS-CoV-2 virus from binding to the sialic acid, and essentially preventing the binding of the virus to the ACE2 receptor as well. So the way that chloroquine binds to the sialic acid, it actually prevents proper binding of the SARS-CoV-2 virus to the ACE2 receptor. So this is another mechanism by which chloroquine and hydroxychloroquine can inhibit the SARS-CoV-2 virus from actually infecting a host cell. So again, the three ways that I talked about in this lesson are that chloroquine and hydroxychloroquine can alkalinize endosomes and lysosomes, preventing endocytosis, so preventing this process from occurring, can also allow the influx of zinc into the host cell. Increased zinc concentrations can lead to inhibition of the RNA-dependent RNA polymerase and preventing viral RNA from being formed. The third point is that chloroquine and hydroxychloroquine can bind to sialic acid, preventing the S protein on SARS-CoV-2 from properly binding to the ACE2 receptor. So these are the few mechanisms by which chloroquine and hydroxychloroquine have been shown to have some antiviral effects, at least in vitro. Again, the big disclaimer here is that all of this has been shown in vitro, essentially in cell culture in a petri dish. So this is far from being shown in human clinical trials. So keep that in mind when we look at this evidence so far. So I hope you found this lesson informative. If you did, please give it a thumbs up. Consider subscribing to the channel. Any and all support is greatly appreciated. And as always, thank you so much for watching, and I hope to see you next time.